much. After the previous presentation, my task is a little bit humbling. It's a small country in the Caribbean Sea. I'm Radjin Steingrover. I'm a clinical microbiologist, as of this year working in Curacao, before in the Netherlands in Europe. And I will present some 5-year data on resistance to antimicrobials. But first, let me give you some perspective. For Curacao, it's a Caribbean perspective. There are many small countries and fewer large ones like Haiti or Cuba. But there are many small islands, many small countries, and problems may be similar, but also very different among them. Curacao, approximately 150,000 inhabitants, 1 hospital and 1 laboratory, and Curacao is part of the former Netherlands Antilles, which are spread across the South and the North Caribbean, and are now separate countries within the Kingdom of the Netherlands. To put it on a map, it looks like this. If you recognize the Caribbean Sea and the islands around it, you find 3 islands just off the coast of Venezuela, down there, and the middle one is the largest one, is Curacao. If you look at the island itself, it has mainly 1 urban area, the large yellow part, which is Willemstad, where most of its inhabitants live. It's a country with a relatively middle income characteristics, I think, while a large part of the population is living in circumstances that are closely to poverty, with low incomes and low facilities like sanitation, etc. If you have seen pictures of Curacao Willemstad, you may recognize this one, which is the traditional waterfront, which dates back to the 17th century, colorful colonial houses. This is the ADC, the Analytical Diagnostic Center, laboratory facility where I work, which houses clinical chemistry, toxicology, and of course the microbiology department. And in more ways than one, Curacao is an excellent place to work and do antimicrobial research, as you can see here. To also give you a perspective on hospital hygiene, and instead of a nice picture of a very old hospital, you see a gray square, so something went wrong. But the text clarifies some. It's an old building, and politely said, it's difficult to keep clean. In fact, it's impossible to keep clean in a modern sense, in a modern perspective, how you think of your hospital to be clean and sanitized, etc. It doesn't have proper isolation facilities, and the building and the materials used make it unsuitable to have it properly sterilized, sanitized, in case of calamities. The people that work there, the medics, they have little or no guidance on antibiotic use. They did an antibiotic guideline in 2012, but if you ask them, hardly anybody is familiar with it, and over 60% of prescriptions are off the guideline. I'm sorry, the graphics are failing here. This was supposed to be a picture of a very important report on the KBC outbreak in 2012, which attracted a lot of attention within the country and from the authorities, the way it was handled. And now, I'm sorry, these were meant to show graphics of that small outbreak in the hospital in 2012. What was seen on the graphics is a rise in incidence of several KBC types in the hospital, mostly clustering to a KBC 2 type and some other types as well. KBC 2 and 3, as were found in this outbreak, are very regionally related to North and South America. So not surprising to find there. Not surprising to find an outbreak of KBC in a country, in a hospital that functions like as I described. The outbreak prompted some actions, beefing up their antimicrobial stewardship practices. One of the consequences that I worked there stems from this outbreak, and indirectly also the results of the analysis that I will show you. The main questions for the analysis in the title of this talk were, well, what is the extent of the antibiotic resistance? Is there any trend in resistance over the last 5 years? Anything to anticipate or anything to guide us in antibiotic use there? Moving to methods of this analysis, we used ADC database, which contains all the cultures of the island. What I will show you is limited to blood cultures, indicative of real infection, serious infection, sepsis. Doubles or multiple samples are filtered according to methodology that is common to the Netherlands and Europe, which is most known in the NETMAP and EARS databases. And it filters down to 1 species per patient per year. So 1 patient can contribute an E. coli and Klebsiella, but 1 patient cannot contribute an E. coli in January and an E. coli in December, which is a limitation of the method, but it gears toward empirical therapies. To make it comparable, I've used the same analysis for curacao, which I will present to you in a few slides. I excluded the most likely contaminants, and this table shows you the main result in numerical. Here are the species listed, and over the years there are numbers contributing to our blood cultures via the method I mentioned. As you can see, Klebsiella pneumoniae, Escherichia coli, the top 2 gram negatives, they are not that different in numerical presence. If you move down to other Enterobacteriaceae, the numbers drop very fast to 20 per year or so, even further more down. Yeah. You get the non-fermenters like Pseudomonas aeruginosa, smaller numbers for Acinetobacter, Stenotrophomonas, approximately 50 Staph aureus in blood cultures. The largest swath is the coagulase negative Staphylococci, which I deemed mostly as contaminants, Enterococci, and various others. Graphically, it's represented like this, where in the various circles, inward to outward, you see the several years from 2010 to 2014 outward, and the large purple section here is the Staph aureus the Staph non-aureus, and those are excluded in this graph, showing you a more informative picture with E. coli, Klebsiella, the other gram negatives, and the gram positives here. Down to the individual species and their susceptibility patterns. You get graphs like this. For Staph aureus, the percentage susceptible is depicted on the y-axis for several years, 2010 in the lighter, 2014 on the darker side, for the antibiotic susceptibility panels that we used, or actually were represented in the database. Quite limited for Staph aureus, but as you can see, flucloxacillin, vancomycin, erythromycin, clindamycin, and cotrimoxazole. Good susceptibility for cotrimoxazole, vancomycin. High percentage susceptible to flucloxacillin, but very worrisome is cursor goes away, is a trend towards a decrease to flucloxacillin susceptibility, which represents an increase in MRSA, and that's a bad thing. Enterococcus, that impresses like more like a wild type distribution, no trends there, no indication of vancomycin resistant enterococci. Moving to the gram negatives, Escherichia coli with a more larger antibiotic panel depicted below. Here you can see the penicillins, ampicillin, amoxicillin clavulanate, cefuroxime, second generation cephalosporin, ceftazidime, cefotaxime, the 2 representative of the third generation cephalosporins, nitrofurantoin, cotrimoxazole, fluoroquinolones, aminoglycosides, and far right the carbapenem, meropenem, showing you 100% susceptibility, but these susceptibility percentages, 80%, around 80% for third generation cephalosporins. That correlates, on the other hand, with approximately 20% resistance or 20% ASBL present in those isolates, which is quite high. For Klebsiella pneumoniae, we're not going to spend so much time on that one, but all the percentages are lower, where you have ASBL up to 20% in E. coli. For Klebsiellas, it's close to 30%. The carbapenemase KPC, present in the years 2012-2013, shows also in the data from the blood cultures, and that shows that it's not only colonization, but it's actual infection as well. Pseudomonas aeruginosa, smaller numbers, more variation in the depicted susceptibility. Ceftazidime, very susceptible over the years, not very good to interpret to be interpretable for a trend. Meropenem, susceptibility is high, and ciproflox sensibility close to 90%. To summarize these results, MRSA, moving towards 10% in blood cultures with an increasing trend. The gram-negatives show high percentages of the ESBLs, and we saw 2012-2013 KPCs. Trends over time, they vary if you noticed. Some are increasing, some decreasing. Don't understand really why that is. I would be cautious to interpret or accept these trends. Meropenem, last resort antibiotic, doesn't have guaranteed efficacy in empirical therapy. Practical consequences for the hospital: frequent isolation of highly resistant microorganisms that require isolation of patients, that require adaptation of therapy. We have circulating KPC strains that are difficult to treat, only susceptible for colistin, amikacin, and some of the newer antibiotics, which are practically difficult in the curacao setting. Moving to implications and goals for antimicrobial stewardship, I would like to present a focus on the beta-lactam antibiotics. As you can see, over time, penicillins, cephalosporins have evolved in several generations, and finally carbapenems. And in their wake, or after their use, the resistance has been selected or emerged. Penicillinases, cephalosporinases, ESBLs, the third generation cephalosporinases, and most recently more carbapenemases. In my experience, or the hospitals, in my experience, antibiotic use follows the trend in resistance. With increasing resistance to these generations of beta-lactams, first-line antibiotic use or major antibiotic use moves away from penicillins and second-generation cephalosporins, more towards third-generation cephalosporins, either with or without aminoglycosides. But if resistance becomes very extensive and very variable, I could see an overuse of antibiotics, overuse of carbapenems, without a proper logical pattern. And I think the curacao situation is more similar to the last slide, to the last graph. So the implications for the goals is basically a return to rational use of antibiotics, whatever that may be. But it's a good question and a good goal. And I'm not defining the details yet, but we'll have to find out what exactly is suitable and rational for this situation. And I think lots of data emerges, and we are learning a lot these days, these years. We have implemented some parts of antimicrobial stewardship. We are updating the antibiotic guideline, and it's the spread of the knowledge of the guideline. we are doing, we are going to start with Precies registration. Precies is a Dutch term, but it is basically a registration for hospital infection to see what's causing diseases in our hospital. Antibiotic usage data is now collected and provided and presented. Resistance data is now available. Hospital hygiene has been reinstated as a department and increased in staff. An antibiotic and infection treatment team will be formed this year, and what I would like to add to the standard stewardship list is that we need local research into the sources and the chain of transmission of HRMOs. And we are doing exactly that starting of this year in Curacao, and the role of sewage and habitation. We are now looking into all sorts of water to find gram-negatives and their resistance genes. So coming back to the happy picture, Curacao people living with their water, we are now looking at it with different eyes. With that, I would like to leave you. I would like to thank the institutions on Curacao, ADC, which is the laboratory, and the SEOS, which is the hospital side. I would like to thank you for your attention. I would like to thank the organization for having me.